I've long since thought that biology should be elevated to the level of astrophysics in our minds, so this is the perfect venue. So you might be wondering why a professor from Wits, from Johannesburg, is here to talk about thicket ecology. And the answer to that is, I know this forum likes to think about animals. So it's like a kudu in the road, a kudu that jumps out into the road in front of you. So Daniel Butner was the kudu. He approached me in, 2020, I think. I'd just come back from overseas and I brought some fun new tools, and somehow Daniel had got wind of this, and so he sent me an email and we started working on thicket physiology. As Alistair says, it's been this microphone's going to bug me. I'll just hold it. As Alistair says, it's been a remarkable journey. It's been quite fun to think about the physiology of this part of the world. So I'm very pleased to be able to share some of the results with you today. So this is a bit of drone footage that Alistair took at Kabucha, I think in August 2022. Alistair can correct me, I made the date up, but it corresponds more or less with when we were working there. And if I play it, hopefully it does play, it's quite large. What you will notice, hopefully, is that firstly, this is a remarkable vegetation type. That should be the first thing that strikes you. But then the second thing is that there are very different responses to the drought. You see some trees that have died off. You see some grey-looking plants. You see some light green plants. And so the question for a physiologist is, what's going on here? Can we make sense of which species are dying during the drought? Can we quantify the extent to which other species are losing function during the drought? And can we use this to predict future drought events, future impacts of future drought events? So that's essentially the basis of what I'm going to communicate to you today. We asked essentially a very simple question: what are the drought impacts on thicket? Can we say something meaningful as physiologists and ecologists about the drought impacts in this remarkable vegetation type? Okay, it's going to result in some neck pain. So to explore that, we've got a very general framework of how plants respond to increasing water deficit. So what I'm going to show you is essentially that conceptual framework. On the x-axis here, you can think of this as time. And then on the y-axis, we've got plant response. I'm going to try stand this side, see if that helps. This is better for an astronomy show. So on the y-axis here, we have plant response. And the first response that happens, much like if you walked out into a desert, your physiology would start to slow down. You would start to maybe decrease your breathing rate. You'd try to conserve energy. Plants are very similar. They essentially close their stomata to conserve water. So the rate of water loss decreases as the drought severity increases or carries on. Stomatal closure is very tightly linked to turgor. So turgor is the water pressure of a cell, and this pressure determines much of the physiology of plant cells. So this is essentially, you can think of this as the level of hydration of the cell, and that governs a lot of the physiological processes that occur within a plant. So this blue section here is where active physiology takes place. That's where all the metabolic processes that rely on water essentially occur within the plant. There's another phase, which is this damaging phase, this phase shown in red here. So once the plant has closed its stomata, they continue to lose moisture because nothing is perfect. So much like even if you found a shady spot in the desert under a tree, you would still need to breathe. You'd still need to essentially lose water. At some critical point, the water column within the plant starts to break down. So you can think of this, you can think of water transport as like a rubber band, being driven by a rubber band. So water is being pulled up by the leaves. It's essentially creating a tension on the water column. If that tension gets too substantial, if it gets too big, if it gets too great, the water column will snap. That's what this gray line here is showing. This is the increase in embolism or air blockages within the xylem, the internal vascular tissue of the plant. This is the degradation or loss of function of the internal water transport system. The arrow here is one-sided. Okay, it's non-recoverable. Once the plants embolize, once air is introduced into the pipes, they don't have a heart, they can't pump water, they can't, much like us, they can't pump blood, they can't overcome a loss of attachment of the water column. Okay, so that's a critical threshold that the plants are trying to avoid during a drought event. Okay, so this is the conceptual framework of how a general plant responds to drought. This is more or less useful in a thicket context, but it doesn't answer our question. It doesn't tell us which species are likely to be impacted more than others. To explore the idea of embolism a little bit further, just to cement this in your mind, I have a video of a dehydrating leaf. Okay, so this happens to be a tomato plant leaf, but it'll work for the purposes of today. And what I'm going to show is essentially two sections of the leaf. We have this blue section and this red section. And what those illustrate is the venation and the leaf tissue that is supplied by specific venation. Okay, so this experiment, what we did is we brought the plants into the lab, we dried them down, we took repeated images of the leaf as the plant dries down. Okay, so what you'll see is that the leaf starts to shrink. At some point, we'll observe embolism in this particular conduit, and then we rehydrated the plant. Okay, and I want you to observe what happens when we rehydrate the plant. So hopefully this plays. Okay, so what you see, this is grayscale here, so this is the extent of the tissue supplied by the particular vasculature. And if you watch very carefully round about now, that very quick red color, what that was when the xylem tissue embolizes, it changes from transporting water to being air-filled. We can detect that change in transmission of light visually. So we can, literally with cameras, we can detect the embolism event. I don't know if I can play that again. Maybe it's... Okay, so again, what we're seeing is the extent of the downstream tissue, the aerial extent. Okay, so it decreases, it shrinks. The red tissue essentially will remain desiccated. Okay, it'll continue to shrink and dry down even further, while the blue tissue, when we rehydrate it, will increase in extent again. It'll recover. Okay, so all this illustrates, hopefully, is just the idea that embolism is non-recoverable. It's a critical threshold beyond which plants cannot recover if they surpass it. Okay, so then in terms of this study, this presentation, I'm also going to introduce another concept, which is that we can quantify those processes. So we can quantify a vulnerability curve, we can quantify the turgor and the change in turgor of the plant. So I'm going to convert the conceptual framework that you saw earlier into this kind of format. This is a traits-based framework for how we think about those generalized responses. So the first thing to notice is that the x-axis has changed. This is now water potential, the water status of the plant, the level of hydration of the organism on the x-axis. And we've still got plant response on the y-axis. I'll just introduce a couple of different components. Okay, so in terms of the turgor-driven processes, we can measure what's called pressure volume curves. We can measure the extent of turgor within the plant. We can measure the level of hydration in the plant. And this point here is known as the turgor loss point, the point at which turgor is lost in the plant. Essentially, it's the end of active physiological processes within the plant. The other process that we can quantify is the process that I showed in that video, which is the xylem vulnerability curve. This is the amount of embolism that is present within the xylem of the plant. We extract a particular metric that I'll refer to as P50, which is just the water potential at 50% loss of function. So if you think of 100 pipes, 50 of them are now filled with air at this particular point. This is a catastrophic level of dehydration for a plant. Okay, so that's also the steepest portion of the curve. And it illustrates the extent of the damage within the plant. Okay, so we went out and, Daniel went out and he measured these vulnerability curves, and these are the results that he found. I'll walk you through what these mean, and I'll do this by thinking about the different species. Okay, so if you focus your attention on the top right here, the first species that I'm going to talk about is Polygula, and what you might notice is that Polygula has very little safety between where it loses turgor and where the red bit starts. Okay, so the xylem vulnerability of Polygula is relatively high. Okay, it's quite vulnerable to drought. The other thing that I want to draw your attention to is the idea that we measured the water status. These two lines here, these 2 points rather, indicate the water status in a wetter year and the water status in the drought year. So we were very fortunate to be able to measure plant physiology during a very dry phase. This was, if you can remember, the 2020 drought. It was a very protracted drought, and as I say, we were very fortunate to be able to measure the physiology while it was happening. And what this results in is the idea that we see a lot of mortality in the Polygula across this particular study site. We worked at Kabucha Farm in the Addo area. So Polygula, we suspect, is a species that is likely to be highly impacted by the drought. Again, as shown by these images. There is one element of Polygula that I'm uncertain about, which is that it could be playing a different life history game. It could be playing a game of growing quickly, being relatively vulnerable, and investing a lot in reproduction, investing a lot in the seed bank. So it could be playing a persistence in the seed bank game. That's something that I think is worth exploring. But in terms of these individuals, there's no chance of recovery. These plants are dead. There's loss of function in these particular individuals. The second species that I'm going to talk about is one that's been commented on earlier in the forum, Papia capensis. Papia is we're starting to get to the bulletproof edge of thicket. Something that's maybe not apparent to you, but is to me, is that these P50 numbers are very resistant. This is an exceptional vegetation type in terms of its capacity to withstand, its physical capacity to withstand embolism. So these curves, Euclea, Cercia, and Papia, they're at the very resistant edge in terms of plants from around the world. So this, in terms of drought tolerance, this might be no surprise to many of you in the room, but thicket is an exceptional vegetation type. We just have the evidence now in terms of its physical capacity to withstand drought. So with Papia, we start to see a larger safety margin, a larger gap between when it loses function in terms of the physiology and when it starts to experience catastrophic loss of function in terms of the xylem. Okay, the other thing to notice with Papia, again, during the drought, the orange cube here, the orange square here, the individual surpassed P50. So we expect severe impact in papaya. And sure enough, when we went out into the field, we quantified a lot of mortality within papaya. This is almost certainly driven by xylem embolism and downstream desiccation of the mesophyll tissue. So what you can see here is a struggling individual. You can see some dead leaves, some living leaves. That's the idea that some of the pipes have already embolized, some of them are still transporting water. If the plant was rehydrated at this point, some of these leaves would survive and the others would remain desiccated. Okay, but for other individuals across the full canopy, the individual experienced dieback. Okay, so this is degradation of one of the bulletproof species within this flora. The last species that I'll focus on is Cercia, and we were quite surprised with Cercia. So Cercia is, again, bulletproof. These numbers are extraordinary. -8 is one of the toughest angiosperm P50s that we've recorded globally. Again, there are only on the order of 14 or so lineages that surpass, 14 genera that surpass these numbers globally. The other thing to notice about Cercia is that its water potential did not reach these catastrophic levels. So we might expect a fully green, fully vibrant canopy when we go out to measure it, because it hasn't experienced these non-recoverable damaging thresholds. Again, large safety margins, right? So we expect that this species is likely to be least impacted during the drought. When we went out there, what we saw was yellowing leaves. We saw canopies that looked as though they were degraded. Okay, and so this was quite a surprise for us. We didn't expect this. And what it reveals is that there's a drought deciduousness element to the vegetation. Again, this might not be surprising to some of you, but it was to me, just in terms of the predictive physiology. So we quantified the impact at two different time points. The one was during the most severe stage of the drought in 2020, and the other was in a recovery phase in 2022. You can see here, this is an index of the severity of the drought. So in 2020, the plants had just gone through essentially this protracted dry period. It hadn't rained for a long time. The rain that had fallen was not substantial. In 2022, the rains had returned. The blue colors here indicate wetter times. The yellow colors indicate drier times. What we tried to capture was the impact of on the vegetation in terms of 4 metrics. Okay, I've tried to illustrate them here. We started with a leaf level assessment of the number of dead leaves across the canopy, so averaged across the canopy. We looked at the discoloration of the leaves. We looked at how many leaves were retained, how many living leaves were retained on each branch, again, averaged across the canopy. And then we looked at canopy condition, the intactness of the full canopy. So the matrices were designed to scale from a leaf level right the way through to kind of a canopy condition level. And I'll just illustrate those 3 species that I mentioned earlier, so Papia, Polygula, and Cercia. If we look at the comparison between Polygula and Papia, what we notice is that during the drought, there's relatively, if we just look at crown extent, there's relatively low crown extent in Papia and in Polygula. In Polygula, that decreases as we enter into the recovery phase. These plants, once they surpass that critical threshold, they desiccate, they die. There are a lot of dead Polygula individuals on the landscape. This also indicates some long-term effect of the drought. During the drought, leaves that looked green may have already have experienced embolism within the xylem, and the leaves themselves may not have desiccated yet, but they would eventually. And I think that's what leads to this decrease following the recovery, the rainfall phase. With papia, a dead individual on the landscape is a dead individual on the landscape later, so there's no recovery essentially of papia with the recovery of rains. If we then look at Cercia, what we notice is that, so Cercia is here, during the drought, there is high leaf discoloration, and that's true of papia as well. In papia's case, the leaves turned brown. In Cercia's case, the leaves were yellow. We think what is happening there is that Cercia is drought deciduous. Deciduousness or leaf senescence is a programmed condition. Essentially, we think that leaf shedding in Cercia is not a damaging response. It's an active physiological response to try and conserve water further. So Cercia is really a bulletproof plant within this landscape. And then the other results that we can add to that. So what we're adding to our general framework, if we zoom back out, is that we know that stomata close, and this varies between species, which I'll discuss shortly. But what we're adding is that there's this active leaf shedding phase that also is an attempt to reduce the effect of embolism, reduce the rates of water loss. And this is something that's, I hope you get a sense of the mixed responses within this particular group of woody trees. For me, it's quite interesting to think about leaf shedding just in the sense that it has to occur, it has to at least be initiated while the plant still has turgor, if it's an active physiological process. So my thinking has changed from originally thinking that leaf shedding is a drought damage response to thinking that leaf shedding is now an active physiological process designed to delay the negative effects of the drought. Okay, so hopefully we've answered our question. We can look at this vegetation type, we can understand the papias, we can understand why they have brown leaves, we can understand the gray shrubs. Hopefully this is a Cercia, right? So the leafless shrub, we can understand that plant will recover following rehydration. This plant won't. We can understand that Polygula might disappear from the system for a while, at least. So we've answered the question of which species are more vulnerable and why, and hopefully we've also captured, to some extent, some quantitative assessment of the damage of the drought. I'll spend a few seconds thinking about drought strategies. So what we think is occurring within the thicket is that there are a couple of drought strategies. There are these live fast, die young individuals at the top end, and then there are these bulletproof individuals at the bottom end. Large safety margins, highly resistant xylem, very impressive drought tolerance. One of the aspects that we haven't considered was, of course, the succulent component of the evergreen succulent thicket. I think that's what it's going to be called. So this is something that I'd love to work on. To what extent are the succulents likely to be affected by droughts? I think it's truly remarkable that we can think about one particular functional type, the woody component of the vegetation. And within that woody component, we see very different, very mixed drought response strategies. That's one functional type, but there are many within this vegetation type in total. So that's something that's remarkably exciting. I just wanted to say that I did grow up in this part of the world, and it's a remarkable vegetation, and it deserves to be explored further. It really is great to see this forum and what you guys are doing. Yeah, I think it's truly remarkable. So with that, I just want to thank Alistair and Daniel for great discussion. Daniel did a lot of the work. He deserves a lot of the credit for this particular study. And then the landowners, I didn't have too many interactions with them, but we received accommodation. They were very generous with allowing us to work on their land. It's it's really good to be able to investigate physiology and physiological responses in a natural setting. So yeah, tremendous thanks to the landowners, the land managers, the people that allowed us to work in their space. And then finally, the institutions that contributed to funding and a workspace. Thank you.